Okay, well, good afternoon. It's a great pleasure to be here, particularly to be here on this occasion of celebrating Govind Khorana. I have this distinct memory as an undergraduate of learning about the experiments of Khorana and Nirenberg and their colleagues, of trying to understand the genetic code and really thinking about this approach of perturbing, the nucleotides that you'd feed into this system of translation, and then to see what proteins come out. And I just remember really appreciating how exhilarating it must have been to put in these different nucleotides and then figure out the principles of this really fundamental code. And I think that's really been an inspiration for me throughout my scientific career of just really being excited by the approach of tinkering to try to understand the meaning of biological information, that is to alter things and then see what comes out, see what biology tells us. So as a faculty member, I've focused my lab on applying really this kind of approach to trying to understand cell signaling and signal transduction. That is to say that cells, one of their most amazing properties is the ability to act as very complex and sophisticated signal processing devices. That is, they're able to detect many different inputs, and then through these complex biochemical circuits, make decisions about whether to grow, divide, to differentiate, to move, and so on. And so we've been interested in trying to understand how is it that systems of signaling proteins work together to become these systems that can process information and make decisions, but also we're not just interested in how any one particular system works, but rather the problem of how do diverse and complex responses evolve, since, as you can imagine, in the constant struggle of evolution, the ability to sense new things and make new decisions is very important. We see a huge diversity of responses in cells. And so how is it that signaling systems are set up that new responses could evolve? So in the early 90s, one of the great breakthroughs in this came from work of people like Tony Pawson and others, where the many signaling proteins that were sequenced, one of the things that came out was the discovery that, at least in eukaryotes, many signaling proteins were made up of modules. So you could recognize, based on conserved sequence motifs, blocks of genetic material that were used over and over again in different proteins. And so this suggested that perhaps these were the evolutionary building blocks for building signaling pathways. And so what we've done more recently is focused on really applying this synthetic biology approach to study and understand the logic of cell signaling pathways, to try to understand how is it that we can use modules to build new signaling responses. So in contrast to sort of more traditional biological approaches where you have a system of interest and then try to deconstruct what are the parts and linkages that give this, what we're interested in complementing this approach with is to also look at it the way that an engineer might, of trying to take parts and understand the logic of how they're put together to build systems, and not just one particular system, but many different systems. And I should point out, this is not just the problem of an engineer, but also the problem that evolution faces. It has these building blocks, and it has to figure out how to build new systems that give you some sort of fitness advantage. So the hope is that by combining these two sorts of approaches, we can get a better understanding of a way of deciphering the basic logic of the meaning of this sort of signaling information, as well as the interesting possibility that we could actually understand and create new novel signaling responses. Okay, so just to give you a brief outline of what I'll talk about, I'm going to just review the basic grammar of how we think signaling modules work, what are the different domain types that they come in, primarily catalytic ones and interaction ones, but then how some of the fundamental rules that we understand about how they can be combined to give you complex behaviors. And then I'll talk about just one really example of how we've tried to build new signaling functions using these kinds of modules, to use this toolkit of modules to make new signaling proteins and new functions. We've done a number of studies on trying to build complex signaling switches that can act as logic gates with many different sorts of operational properties. I'm not going to talk about that. Instead, I'm going to focus on this one case of trying to use scaffold proteins as a way to rewire kinase networks. Okay, so as I said, there are these different sorts of modules that you find repeated in signaling proteins, and one type, one general class, are the catalytic modules that most of us think about, the ones that actually transmit information. So these are things like kinase domains or phosphatase domains, and these transmit information by changing the state of some target protein between, in this case, a phosphorylated and unphosphorylated state, and so these act as the writer and eraser of these change of state marks. And this is common to almost all signaling systems. If you look at like GTPases, you have a toggling between two different states, a GDP and a GTP bound state. And again, there's a writer enzyme, the guanine nucleotide change factors, and then sets of eraser enzymes or gaps that reverse the reaction. And in many cases, but not all, as Mark Potashi pointed out, there are some reader domains, like in the case of phosphotyrosine, SH2 domains that act to recognize those things. So you have these sort of modular functions. And what we find if we look at these things at signaling pathways is that these sets of information processing modules are linked together in diverse ways. They can be linked together to form cascades, feedback circuits, the things that give you the complex behaviors that we see. Okay. Now, the other class of modules, major class, are ones that are involved in interactions. And these really act more to direct and control the flow of information. That is, oftentimes we'll see a catalytic domain like a kinase combined with these other domains, which are, for the most part, protein-protein interaction domains. Many of them, for example, the SH3 domain, whose structure is shown here, recognize short cognate linear motifs, in this case, proline-rich motifs. So how do these domains work together to give you a new function? So I'm just going to outline two of what I think are really fundamental ways in which you can combine these sorts of modules to give complex function. One is just through the simple idea of recruitment or co-localization, that in many cases, for example, if you have a kinase that has to interact with a specific target, you will find an interaction domain linked to that kinase and then the cognate partner in the correct partner. There are higher order or more complex systems that use this sort of mechanism. For example, and I'll talk about this in more detail, there are scaffold proteins that are essentially arrays of multiple interaction modules that can act to link together sets of proteins that, for example, might form one pathway. Okay. Now, another major way in which these domains can be combined is through allosteric regulation. So oftentimes you'll have a catalytic domain, say like a kinase, and it will have other accessory domains like these interaction domains. But what you'll see is that they can exist in an off state where, for example, this domain might occlude or somehow inhibit the function of that catalytic domain, or participate in an intramolecular interaction that's, for example, conformationally locks it in an off state. But in this way, either of these cases, addition or binding of something that displaces this interaction can act as a mechanism to release this and then give you activation. So these, of course, are not mutually exclusive. There are many cases where these sorts of domains both play a role in controlling and shutting off activity, but then recruiting them to the right sites and then also activating them. So I'm going to just focus on that first case and tell you about some work that we've been doing for a number of years on scaffold proteins, which really are a modular platform for wiring kinase pathways. So obviously, one of the classic problems that a cell faces in trying to process information is the abundance of all the different types of information that it's dealing with. The cell is bombarded with information, much like this switchboard operator, and she has to keep everything straight. And not only is there a lot of information coming in, but there are a lot of signaling proteins, even protein kinases in the human genome. There are about 500 different protein kinases, many of which are closely related. So one concept that has emerged, as I've told you, is the idea that scaffolds can play a role, essentially acting as a molecular circuit board that helps to decide what kinases or what signaling proteins will communicate with one another in a specific way. So there's the idea that scaffolds may help determine pathway connectivity. In addition, more and more we're finding that scaffolds don't just connect, say, kinases that play a sort of have a linear relationship, but that they could also recruit both a combination of positive and negative factors and do so in a temporally regulated way. And so that by having things that modulate the pathway, they can shape the response through things like feedback and other mechanisms. In addition, of course, one of the really attractive ideas about scaffolds is that they may be a platform that helps in evolvability. That is, one simple way to imagine evolving new responses is simply by changing scaffolds, and then you could use then the same sets of catalytic enzymes then might be able to be used to give you new types of input-output behaviors. Okay, so to look at and explore these different hypotheses, what we've tried to do is to test them by trying to rewire pathways by altering the scaffold. Can we change function? Can we get new function by changing these interactions? Okay, so the system that we've been working on primarily is the yeast mating pathway, where there's a protein called Ste5 that plays a role of the scaffold. So in the mating pathway, this signal is transduced to a number of different transcriptional and other changes via this MAP kinase cascade, these three kinases that successively phosphorylate and activate one another, sterol11, sterol7, and fuz3. But quite a few years ago, it was discovered that there was this additional protein, sterol5, that was required for this function, absolutely required to give you signal transduction. It was found to bind all three key members of the pathway, and the model emerged that it was important for properly directing signaling in this pathway because, like all eukaryotes, including ourselves, yeast have several different MAP kinase pathways linked to very different responses, and it's important to keep those straight. And in fact, in particular, several of these components, the kinases here that are involved in the mating response, are actually also used in distinct signaling pathways. So let me tell you about how we've tried to look at whether we could get new input-output connections by changing the scaffold. And so this is work that was started a number of years ago by a former postdoc, Sang-Hyun Park, who was also a graduate student here with Ron Rains. But as I told you before, we have this complex that's associated with the mating response where pheromone leads to this mating response. But there are other MAP kinase pathways, including the osmolarity response pathway, where high osmolarity stress will lead to various responses. And this is also mediated by a MAP kinase cascade. Now, it's thought that this is assembled by another scaffold protein called PBS2. One thing I want to point out about these two pathways is that they normally, under most conditions, do not communicate with one another. They do not crosstalk, despite the fact that they are both MAP kinase pathways. But one thing that's particularly striking is that they actually share one component. The MAP3 case, sterol11, is the same molecule in each of these, yet you don't get this crosstalk. So the hypothesis was that the population of C11 associated with this complex is distinct from the one in this case, and it knows who it was activated by and who it should then communicate with downstream. And that's why you have these distinct physiological pathways, because you have distinct complexes. So Sang-Hyun reasoned, was it possible to actually try to make new pathways by combining parts of these scaffolds to, for example, generate a yeast cell that would, when stimulated with pheromone, give you the osmolarity response. So we did a very simple thing, which was to fuse these two scaffolds and then make deletions that would knock out things involved in transmitting the downstream mating output as well as receiving the upstream osmolarity output. And what he found, in fact, was that this cell bearing this diverting scaffold actually very effectively and very specifically took the pheromone signal and translated it to the complete osmolarity response. So in fact, he could essentially evolve or do forward evolution of a novel pathway by changing the scaffold. Just one other thing that he did was to show that heterologous interactions could also rescue signaling. Jeremy Thorner's lab had identified point mutations that knock out binding of one player in this pathway, and these don't work. So what Sang-Hyun also did was to ask, could we re-recruit those missing kinases using a completely different orthogonal interaction, in this case a PDZ domain interaction that plays a role in the mouse neuromuscular junction, so it has nothing to do with yeast mating. And this just shows the mating efficiency of the two mutant sterol5s with those point mutations. They're completely inactive. But then if we rescue with these two PDZ interactions, you see that you do get, in fact, very significant rescue. So we can, in fact, with very unrelated interactions, stereochemically very distinct ones, still regain this pathway function. One thing I do want to point out is that you can see that this is still several orders of magnitude lower than what we see in wild type. So in fact, just to sort of elaborate on the point that Mark brought up in his talk earlier, is that the way that I sort of look at it is that these kinds of tethering interactions probably play a really major role in starting a new pathway, sort of a starting point in evolution, where we get a completely new function. But that, you know, obviously living systems work to optimize these things, and in cases like disease, the details of this kind of difference are very important. So in fact, in this case, we've more recently shown that there are very interesting higher order mechanisms that are layered on top of this tethering that we published recently in Cell that help play a role in determining the sort of the very high efficiency signaling that we see in the wild type case. But again, I think that still, the fact that you can, with these sorts of simple tethering changes, get such dramatic differences or rescue does illustrate that these are probably a very powerful mechanism to at least start down the path of evolving a new pathway. I want to turn and talk to you about a different issue, which is tuning pathway response dynamics. That when we look at signaling pathways, we're not just interested in how an input leads to some qualitative output, but rather that when we look at a detailed dynamics response of a pathway, oftentimes we see very different behaviors. If something it needs to be fast, you see a response that's very fast. If there are responses that are either somewhat deleterious or that are very expensive, you might see a delay where the cell waits till it really knows this is a real input before it turns on. There are other cases where you see things like adaptation, where you see a very quick response, but the system adjusts itself either to turn off something that would be bad if it was on for too long, or to allow you to desensitize and then respond to further increases, as is seen in many sensory systems like vision and chemotaxis. Now, how do you get these sort of diversity of behaviors? What we think is that feedback loops layered on top of these linear pathways play an important role. And so what we've asked more recently is, can we manipulate pathways using the scaffold as a platform to precisely tune dynamics? And so again, the idea here is to use this scaffold as a circuit board to try to now modulate the dynamics of pathways. And so Caleb Basher, a student in the lab, decided to try to do this by asking whether we could bring new factors to the scaffold to modulate the pathway response. And so he wanted to have a new interaction site on the scaffold, and so what he turned to was an orthogonal interaction, a leucine zipper, heterodimer pair. This doesn't cross-react with other yeast proteins. It has a high affinity, about 10 nanomolar. And Charles Vincent's lab at the NIH had worked out a set of homologous zippers that span a range of tunable affinities. So what Caleb did is he put one partner fused it to sterol 5, the scaffold, and then asked, could we fuse the other partner to either negative factors like this protein MSG5, which is a MAP kinase phosphatase, or this protein Ste5, which is a positive regulator. I'll just really focus on this negative one, the phosphatase. So this one dephosphorylates and inactivates the MAP kinase. So what's shown here is the pathway output. This is a transcriptional output of a mating responsive promoter. And this is what happens when you don't stimulate, and then if you stimulate with alpha factors, so you get this big response. If we now put in this negative factor MSG5, we express another copy, and then we do it with a defective zipper, we see very little effect. But if we now put a zipper on it that will bring it to sterol 5, we see that this now completely shuts down the activation of the pathway. Okay, so we can tune the pathway up or down by plugging in new factors to this scaffold. So then Caleb asked whether he could build simple positive and negative feedback loops. And the way he did this was to simply express this synthetic effector from a mating pathway-induced promoter, so that you get the response. It would turn on the reporter, but also turn on this gene, so that now you'd express this negative regulator that would be recruited to the scaffold and, in principle, shut it down. And in fact, that's what we see. That what's shown here is a time course with transcriptional output here. Black is the wild type. It goes up and stays up within this time frame. But if we put in that engineered negative feedback loop, for example, you see it first goes up, but then automatically will shut itself down and settle to a lower steady state. So we can build simple positive or negative feedback loops. Just want to show you that the diversity, we've gone on to show that using this simple set of components, we can really build a lot of diverse circuits, and one of them is this pulse-like response or an adaptive response. Here we've employed a decoy interaction. So in binding interactions, one powerful thing you can do is have competing interactions. And so what we've done is we've expressed constitutively a decoy high-affinity leucine zipper that looks like the one that's on the scaffold. The idea is that if we now turn on the pathway, we will start expressing this effector, but that it will first be recruited to this decoy complex, which has higher affinity. But then once that's saturated, this will essentially act as a sink. It will now be able to bind to this scaffold and exert its effect. And so it essentially would be like putting in a delay before the onset of the negative feedback. So in fact, that's what we see. In gray, you see the response when you don't have this decoy, but as we express constitutively again, increasing amounts of this high affinity decoy sink, that we get a longer period where that cell tracks the wild type response, but then after that period, it will then settle back into the adaptation. So we can get complex behaviors like this sort of pulse generation, and we've been able to use this same simple set of simple toolkit to get circuits that show delay, accelerators, or act as ultrasensitive switches. And I think this is also important because MAP kinase pathways are used in many cases in eukaryotic biology, but we see very diverse kinds of dynamic properties, and we can essentially recapitulate a lot of those with this one same pathway by introducing these new synthetic feedback loops layered on top of the original pathway. So just to summarize, there really does seem to be, at least at a moderate resolution, a fairly simple modular logic of how signaling systems or proteins and pathways are put together, and that we can radically alter cell behavior with simple module recombination, something that I think is a great advantage for both the process of evolution and evolvability, as well as our goals in engineering things. With respect to engineering cell signaling, this is still very early days, but I think it really offers the potential of both reaching a deeper understanding of complex function and how signaling systems work, but also how evolution takes place. And then there's also the possibility of very diverse applications where can we change or program cells to execute useful tasks like both in many areas like therapeutics, energy, et cetera. I just want to touch on some of the directions that we're moving in. One of the things that we're very, very interested in doing is really using more combinatorial approaches with these modules to explore a sort of network topology and function relationships. That is, can we take some target function like adaptation, where an input will lead to a transient but self-adjusting response, and ask, what are not just how does one example do it, but rather what's the whole space of solutions that can achieve that function. So we're pursuing this both in silico and in vivo experimentally, but in silico, working with my colleague Chao Tang, we, for example, just to give you a flavor of this, taken a simple system computationally of three node networks, of which there are 16,000 possible network topologies, and asked if we put in 10,000 parameters per each of those networks, which ones are most robust for achieving this function. And we actually see it's a surprisingly low number, and this is coming out in Cell later this month. We've also been working on combinatorial ways of taking these different modules, these different domains, different promoters, and linking them in a combinatorial way so that we can actually search for function in this sort of way. And I think it's going to be really powerful for both defining what are and organizing what are different ways of achieving some function, but also for looking for engineering functions for different applications. Let me just touch on that on my last slide, which is we're also interested in these sort of more higher order challenge applications, and one of the things that we're interested in among several is whether we can actually engineer the signaling in cells to engineer what could be called smart cell therapeutics. So living cells like this neutrophil are able to achieve very complex functions like the ability to detect a bacterial infection and chase and phagocytose the bacteria. And wouldn't it be amazing if we could understand and how the sensors and decision-making signaling circuits and response circuits are, how those are built and how they can be relinked in different ways. And if we could, in fact, take the toolkit that's out there and learn how to link them in different ways, could we design cells that could find, label, or kill tumors, that could, in a more intelligent and local way, detect and suppress immune responses, as well as transplant rejection, or could we we do things like have the ability to steer stem cells or neurons to precise sites for regeneration? Can we layer on top of cells new function that could now allow them to be to carry out new functions, but also they could be things that were more for safety, as was touched upon in the previous talk. So let me end there and just thank the people who did the work. The work on the MAP kinase scaffold engineering was really primarily done, again, by Sang-Hyun Park and Caleb Basher. Thank you.